Okay, for introduction, alkaloids are found primarily in plants and especially common in certain families of flowering plants. In fact, as many as 1/4 of higher plants are estimated to contain alkaloid, of which several thousand different types have been identified. So the function of alkaloid in plants is not yet understood. It has been suggested that they are simply waste product of plants, metabolic process, but evidence suggests that they may serve specific biological function. For example, to protect some plant from destruction by certain insect. Next, the chemical structure of alkaloids are extremely variable. Generally, an alkaloid contains at least one nitrogen atom in amine type structure. In example, one derived from ammonia by replacing the hydrogen atom with hydrogen carbon group called hydrocarbon. This or another nitrogen can be active as base in acid-base reaction. The name alkaloid was originally applied to the substance because, like inorganic alkali, they react with acid to form salt. So most alkaloids have one or more their nitrogen atom as part of the ring of atom, frequently called a cyclic system. And alkaloid is also tend to have bitter-like taste.
As for the objective of this experiment, so the first objective, we have to detect the presence of alkaloid in our collected sample, and the second objective would be to detect the changes in Wagner and Mayer reagent between the samples. For the methodology part, we started off with grinding 5 grams of the fresh samples. 10 milliliter of the chloroform was added while the sample was ground. Next, 10 milliliter of ammoniacal chloroform was added and mixed thoroughly. The sample was filtered using Whatman number 1. When the chloroform layer is separated, the remaining extract is transferred into test tube. 20 drops of 2 mol H2SO4 was added and mixed well. The sample will be observed for 5 minutes to see if there was any traces of chloroform layer. The aqueous layer was needed for alkaloid tests. Equal amount of extract is placed in 3 different test tubes. Each were for different tests. Label A for Mayer reagent added, label B for Wagner reagent added, and C is for the control.
When Mayer reagent is added into brassicagent, the solution turns slightly cloudy but not white precipitate form. When Wagner reagent is added into brassicagent, the solution turns slightly cloudy but not brown deposit form. Meanwhile, when Mayer reagent is added into carica papaya, the solution turns cloudy but not white precipitate form. When Wagner reagent is added into carica papaya, the solution turns cloudy with a little brown deposit form.
Next, for discussion. First, for Brassica juncea, as we can see here, for the Mayer reagent test, the solution turns slightly cloudy but no white precipitate form. This indicates the absence of white precipitate suggests that alkaloid is present but in a very low concentration. Meanwhile, for the Wagner reagent test, the solution turns slightly cloudy but no brown precipitate form. This is similar to the result with the Mayer's reagent. The lack of brown precipitate indicates a low presence of alkaloids. Next is for papaya. For the Mayer reagent test, the solution turns cloudy but no white precipitate form. So this shows the cloudiness. Some reactions suggest the possible presence of alkaloid, but the lack of white precipitate implies that the concentration might be low. Meanwhile, for the Wagner reagent test, the solution turned cloudy with a little brown precipitate form. This shows that the formation of brown precipitate, although minimal, it indicates a positive reaction for alkaloid. This suggests that The papaya contains alkaloids that react with Wagner reagent, confirming their presence to some extent.
So in conclusion, this experiment is to find the presence of alkaloid in both collected samples, which are Brassica juncea and Carica papaya. Based on the result obtained, we can conclude that alkaloids are present in both of the samples since it becomes cloudy when tested in Mayer reagent and it produces brown deposit when tested with Wagner reagent.